Hi everyone, welcome back to another video. So this video is on protecting groups, and most importantly it's going to be a mix of strategy from a competitive examination perspective and from a research perspective as well, and this will be clear in just a bit, like how I'm going to be combining all these three together in this particular video. Now if I talk about protecting groups, I am guessing that you are not a beginner in organic chemistry and you have some idea about it, right? And why do we need protecting groups? Primarily because, we need to, as the name suggests, avoid reaction of certain groups. And we cannot really do that because of the issue of chemoselectivity. For example, if I talk about like the pain points in organic chemistry, that is carbohydrate chemistry. Okay, if I talk about carbohydrate chemistry or even peptide chemistry for that matter. Now over here, the concept of protecting and deprotecting groups is very, very important, because if you look at the carbohydrate chemistry, we have mostly primary or secondary hydroxyl groups, right? We are dealing with primary and secondary hydroxyl groups. And you have to, and we need to be somehow selective. How do we, selectively react between two primary hydroxyl groups? Because the difference between their, you can say, polarities would be very, very or reactivities rather, is going to be very, very minute, right? And this is where you would see there's a lot of progress being made in electroorganic chemistry. Okay, this is also another promising field, electroorganic chemistry, where we use current in order to reduce or oxidize certain functional groups over others, okay, or even between the functional groups as well. And similarly in peptide chemistry, as you can understand, we have we have several amine groups, we have several carboxylic groups, and again the reactivity difference between them is very, very minute. So we need these protecting groups in order to react certain groups over others, right? So the basic aim is quite straightforward, that we need to have a group that gets easily attached and then that gets easily detached as well, because we need to remove that group also. So the protecting group should be easily attached and should be later on easily detached. Like basically mild conditions should be required. And then second is that versatility. What I mean by versatility is like what kind of conditions does it get deprotected in? Okay, for example, if it is a very, very exclusive condition, okay, or if it is a very exclusive deprotecting group, in the sense that most deprotecting groups only get deprotected in acidic or basic conditions, right? But if we have a very exclusive group, in the sense that a protecting group which gets deprotected only in very, very specific conditions, then that is great, because then it can be utilized for a lot of reactions, right? And then we have I'll tell you what this get rid means. So I was saying from a research perspective, because if any one of you is interested in research, and this is a very fascinating concept of chemistry, protecting and deprotecting groups. So the objective over here is quite straightforward if you want to do research in this field. Okay, so let's say for any particular chemistry or any particular reaction or any particular functional group, let's say there is a protecting group that is available. So either you can improve upon that protecting group, that means you can develop or you can find or discover a new protecting group which gets easily attached than the one that is already present, right? So let's say there is a protecting group that is already there in literature, which everyone is very familiar, which is being used in industries. But if you are able to develop another protecting group, okay, which has a better attachment and detachment, basically it is easier to attach and easier to detach, then that is a significant improvement. That could be a new form of research. Second is, like I said, improved versatility, that you can find a protecting group that is stable in a lot of different conditions. So let's say there's already a protecting group that is available in literature, but basically it gets easily attached, it gets easily detached, but it does not have a lot of versatility. So let's say that it gets deprotected in acidic conditions, but there are certain reactions which one might want to do in acidic conditions as well. In that case, the protecting group cannot be used. So if you can find a protecting group that is, let's say, stable in acidic and basic conditions, okay, and at the same time it is easily attached and detached, well, that could be a significant improvement, and again something that you could do research on. And the third and the most important is that if you can find a way to do that reaction without the use of protecting groups, that is going to be the best. So that's what I meant, get rid. That means find a reaction, okay, where a protecting and deprotecting groups are required, and develop something in which develop a reaction which basically bypasses this protecting and protecting group, right? So if you can develop that, again, amazing form of research, which would be of very high utility. So from a research perspective, anyone wants to do further research in organic chemistry, this is a very fascinating field. Okay, so now that we have understood a bit about the protecting and deprotecting groups, it's not always necessary that it is used for selectivity purposes. For example, let's say if I have let's say aniline. Now if I want to do, let's say it's bromination, okay, if I want to do bromination of aniline, so I'll use your acetic acid and bromine. Now the problem is that your in this case the bromination will not be selective. Let's say I want to do monobromination, but in this case what will happen is, because of extreme reactivity, monobromination will not take place. So what could be done, or what is actually done, is that first this NH2 is reacted with your simply ACOH or acetic acid, okay, it is acetylated. Once it is acetylated, you can understand that now from an amine it becomes an amide, okay. So now this becomes an amide, okay, NHCOCH3. So this becomes an amide, and because of that now its nucleophilicity has reduced. Over here it was having high nucleophilicity, over here its lone pairs are in resonance with this because it's an amide, so its lone pairs are not available, and then because of that, now if you do your bromination, okay, now if you carry out bromination, now you will have major product with bromination at the para position, okay. So you will have bromination at the para position, and then further you can just deprotect it in concentrated HCl, and very harsh conditions basically are required. But this particular molecule over here, the molecule that we have like I have just noted down over here, that molecule can basically withstand harsh conditions, okay. This molecule, because of its aromatic nature, okay, generally aromatic molecules are very stable, this can withstand these very harsh conditions which are required for deprotection of amide. But if you want to simply deprotect amide for, let's say, sensitive substrates, okay, some substrates that are base sensitive for that matter, or acid sensitive, in that case you will have difficulty in deprotecting the amide group. So that is why the amide protection is not generally carried out, because deprotecting it requires a lot of harsh conditions. Otherwise, if you think about it, if you want to protect an amine RNH2, if you want to protect it, simply if you like have an acetyl, like if you have an acyl chloride for that matter, this can lead to simple, you know, formation of this what do you call, yeah, this can lead to simple formation of amide, and then you can carry out the reaction, and then like this reaction is very simple to attach. But if you want to detach over here, detachment is going to be very, very difficult, because you need very harsh conditions. I think a concentrated NOH and a very, very long reaction time is required. Now before we talk about this example from a competitive exam, let's see another important transformation. Now in this example, a lot of concepts are going to be tested, and that's why I thought this is an interesting example to discuss. So you want this particular transformation, that is, let's see the changes over here. You have a paramethoxybenzyl group attached, which is again a protecting group for alcohols. The acetyl group over here has hydrolyzed, and you have a primary alcohol and you have a secondary alcohol, and there's another alcohol that is present over here which is already protected, okay. So this is the transformation that needs to be carried out. At the outset it looks to be very simple, but let's break it down. So first thing, as you can see over here, is that if we hydrolyze this acetyl group over here, we will get a secondary alcohol, okay, and we'll get a primary alcohol. And if we want to protect this alcohol, as you can see, it has been protected by your paramethoxybenzyl group. So how do we protect this? Basically, this is the group that this OH is going to react with, okay. Let me just use another color. Yeah, okay. So this is the group that we need to react with in order to get this para-methoxybenzyl. So obviously, like I said, if we hydrolyze this acetyl group over here and we get a secondary and primary alcohol, this is going to compete with this alcohol over here, and you will get a mixture of product. But we want selectivity. So obviously the deprotection or the hydrolysis of the acetyl will come later. So if we add a simple base over here, okay, if we add a simple base, this is going to react over here, simple nucleophilic substitution reaction, and you will get your para-methoxybenzyl group. Now the next step is your hydrolysis. So once that is done, then I can do the hydrolysis of this acetyl group and I will get the free OH groups. And I have to obviously add a base which is such that it does not deprotect the silyl group, because in presence of certain bases, this silyl ether group can get deprotected. Okay, so I have to be very, very careful over here that whatever base I'm going to use, that should not deprotect over here. So once that is done, let me just write this whole thing as R over here. Okay, so I've done the hydrolysis, and again secondary will not lead to deprotection of this. Now as you can see over here in the final product that we require, the secondary OH over here has been protected, but the primary OH has not been protected, and this is very difficult to carry out. So what authors have done is they have protected this primary alcohol first. Okay, so how can you selectively protect the primary alcohol? You can use a very bulky protecting group, and they have used this particular group, that is tertiary butyl COCl. So this is the group they have used to protect this OH over here, right? So again, so you can like, this is a simple reaction. If you want, you can add a base, and this is going to attack over here, and this is going to replace the chloride because of the bulky nature. Okay, the secondary alcohol is not able to react, the primary alcohol reacts, and now we can do this protection. Now we have this final group with this OCO. Okay, I'll write down over here OCO tertiary butyl. So now that the primary hydroxyl has been protected, then you can protect the secondary alcohol. Now the question is that why this particular protection was needed. Okay, because you have to think in front, because now if I require a primary OH group, okay, if I did a silyl protection over here and then I use a certain reagent to deprotect the silyl group, then this will also get deprotected and this will also get deprotected. So I have to use a reagent which let's say only deprotects this but does not deprotect the silyl groups. Okay, like you have to think ahead also, and you have to think from the start of the reaction, because if you thought, like, let's say I will use certain protecting group like this over here, then you would have faced a lot of problems along the path. Okay, so you have to be very, very careful. You have to plan the whole synthesis properly, and you have to keep everything in consideration. Now I can simply use is dibyl H. Okay, that is a reducing group, because this is nothing but this is like an ester. So this ester can be reduced to an alcohol with the help of your dibyl group. So I'll just simply use dibyl H over here, and what dibyl H will do is it will basically simply reduce this group, and I will get the OH back. So my final product will be what I really wanted, which is OPMB this, then this aryl group is protected. Okay, SiPr3 isopropyl. Okay, and then we have your OH. This is a very, very good example of why such protecting and deprotecting groups is very important. And if you ask questions, that is a very, very good way of learning organic chemistry. So let us come back to the example that I was discussing from a competitive exam point of view. So in general, if you want to improve your organic chemistry and also understand questions properly, understanding questions means question that why such and such thing is done, because that will improve your knowledge immensely. For example, this looks to be a very straightforward question, and this question was asked in your GATE 2014 examination. It's related to protecting and deprotecting protecting groups basically. So over here you can see that they have added methanol and H2SO4 in the first step. So what this is going to do, this is simply going to convert your acid into the ester, corresponding ester, right? So this is going to happen. So this is going to be formed. Okay, this is going to be formed. Okay, this is basically a protecting group for the amines. And what is the structure of this? This is simply CbZCl not really sure about the nomenclature, but this is the structure CbZCl. So as you can see, it has a electrophilic center, and this OH over here is not that nucleophilic, because if you see this OH over here, it's not nucleophilic because its lone pairs are in conjugation with this phenyl ring. Okay, and this does not have a sufficiently nucleophilic group present, so this NH2 selectively gets protected, okay, or basically reacts with this and gets protected, right? So we have this your CBZ group attached. So basically you can see over here it leads to a formation of amide, but it is not a typical amide because this CBZ group gets deprotected in the presence of H2 palladium carbon. Okay, it gets deprotected in that case. And how does it get deprotected? First, this, like, okay, let me just quickly show it to you. Like if we have RNH, and let's say this is protected by the CBZ group, which is nothing but this, right? The phenyl group is there. Okay, so then if we add H2PD, first this phenyl group gets cleaved. Okay, this gets cleaved, and then your CO2 gets removed. So this is why it is not like a typical amide, because in the previous in the starting of the video I told you that your amides basically get are difficult to deprotect, but this is not a typical amide. Okay, it forms amide bond, but it's not a typical amide because this group gets removed. The phenyl group over here gets removed easily. Not the phenyl group, sorry. I mean, there's a like, it's not a phenyl exactly. It's like a, it's what do you call it? A toluene, right? Yeah. So it's like phenyl, and there's another CH3 group, right? So there's another bond over here. So this group gets deprotected, okay, and along with that we have CO2 eliminate, and that's how you get the amine. So it's comparatively easier to deprotect than a typical amide. Okay, so the question was that why are we how do we have to question? Okay, I'm not teaching you how to question as such, but like I just want you to be curious as to why something is happening. Now the question is whether this first step is essential or not. Like, can we firstly do CBZ direct protection in the presence of acid? So there's a clear example of this. I have taken from 2 research papers. As you can see over here, this particular molecule, first here they have done CBZ protection and the acid is free. So wherever the question has been posed from, maybe there the authors might have done this protection first. Okay, of not protection, but basically conversion of this acid into the ester first. But over here that is not necessary, because here as you can see that simply first they have done this protection of this amine using CBZ, and then they have done the conversion of your acid into the corresponding ester, and at the same time, as you can see, what they have done, they have added K2CO3 and methyl iodide. This base is very mild, K2CO3. This is again a very good example, because if you use a stronger base, then this NH proton that is available over here, that NH proton will get the deprotonation will take place, and then this N minus can also attack the and get methylated, right? So that is also a possibility. But if you use a mild base, you know that the OH proton is much more acidic as compared to the NH proton, and plus this OH proton, once it gets deprotected, sorry, not deprotected, I'm using that term, once it gets deprotonated, it is in conjugation with this group, so it's highly stable. And similarly over here also, this OH proton, once it gets deprotonated, this phenoxide ion is also very stable. So in presence of mild base, this will form O minus, this will form O minus. They both will get they both will react with your methyl iodide and lead to your alkylation in both the cases. But over here this is not going to happen. Plus that the lone pair that is present over here, it is in conjugation with this CBZ group, so the lone pair is also not available, right? So that is why this can also be done. So it's not necessary that you need to do the esterification of the acid first. Here as you can see, there's a typical example of where CBZ protection was carried out first. And then let's look at the second example over here, again from a research paper that I have pulled out. And as you can see over here, in this particular case, I wanted to help you understand that why this protection is important, okay, that why this group needs to be protected. So as you can see in this case, what they have done is they wanted to react the OH group over here. Okay, they want to react the OH group, but till the time NH2 group is there will always be a problem of competition. Okay, there will always be a competing reaction. NH2 group is known to be very problematic because it's highly nucleophilic, right? So it's always very, very important that if you don't want the NH to interfere with the reactions, to reduce its reactivity, and that is what exactly has been done, and that is why this CBZ protection is very, very important. So anyway, this has been a long video, but I really hope that you enjoyed it and you learned something new. If you did like the content, do not forget to give this video a big thumbs up, and also do not forget to subscribe the channel. It helps reach more people and it gives me a sort of motivation. So again, thank you so much for watching. I'll see you in the next video very soon.